Is it a puppet? I know it's a lion, but I mean, what's its other function? Oh, pencil holder. Okay. So you had one, or? Okay, all right. Now I know where to go if anybody forgets their pencils. Okay, let's go ahead and get started then. All right, so we're on our last 4 topics, 3 long ones and a short one. All right, and so this is number 13, calcium homeostasis. Calcium homeostasis is about calcium, but it's also about phosphate ions in the bloodstream as well. It involves 3 hormones, and then we'll go over the hormones and how they interact to control calcium in the bloodstream. And then we'll touch real briefly on osteoporosis. So for this topic, we're going to consider bones. You probably never really considered bones much, but if you were a dog, you might. So the mineral content of a bone is a mineral we call hydroxylapatite with that chemical formula. Notice it contains calcium and phosphate. However, the mass of bone is mostly living cells and collagen, protein collagen. Now, for most of us, our introduction to the skeleton was one of these things, sitting in the corner of the biology class. And if you were like us, we sort of spent the year speculating on whether it was an actual skeleton or made of plastic. When I was in school, in high school, it was probably an actual skeleton. For you guys, it's probably plastic. And from that view, we get the idea, the thing sits in the corner the whole year and collects dust. We start to think of bone as a static tissue, essentially a structural piece of stone that holds everything else together. But that couldn't be further from the truth. Bone's not a static tissue. And if you think about it, it's obvious. It has to grow. It also, if it's broken, it has to heal. But it also undergoes a process called remodeling, which is a continuous process of formation and resorption. Physiologically, bone, probably maybe its most important function, is to act as a reservoir of calcium and phosphate to ensure the levels of extracellular fluid, calcium and phosphate. So bone is a dynamic tissue. The cell types in it are osteocytes, and these guys in here, so this is a cutaway of a chunk of bone. These guys are the osteocytes. Okay? They exist inside the bone. They're about 90% of the total cells in the bone are osteocytes. And these blue lines are supposed to be the collagen matrix in which these cells exist, and they actually excrete collagen and make most of the structure of the bone. On the surface here, there are osteoblasts. Okay, and osteoblasts, you'll see why this is important in a second, blast with a B. Okay, and those are the cells that build the bone in the remodeling. In little colonies with the osteoblasts are the osteoclasts, which are a big multinucleated cell. And the osteoclasts are responsible for resorption of minerals in bone. Okay. So together, it's the osteoblasts and the osteoclasts that are responsible for bone remodeling. And they do this by taking calcium and phosphate from the extracellular fluid. The osteoblasts put them into the bone to cause formation, and it's the osteoclast that dissolves that mineral to resorb it back out into the extracellular fluid. These little colonies, there's more than 1 million of them around an average adult skeleton. And they're active enough so that on average, we'll all get a brand new skeleton every 10 years. Okay. Calcium is a critical second messenger. In the extracellular fluid, the calcium concentration must be pretty constant. Okay. So one figure you'll see in textbooks is 2.1 to 2.6 millimolar total calcium in the extracellular fluid. However, for our purposes, that's not quite right, because about half of that is going to be bound to protein. So it's not really free calcium. So the free calcium is 1.1 to 1.3 millimolar. Okay, and those are numbers you should memorize. Now, as we've gone over before, we've looked at calcium inside cells pretty regularly. Inside a cell, the calcium concentration is very tightly regulated. Okay, and hopefully you recall something like this. There's calcium channels, there's the calcium release channel for intercellular stores. Outside, it's 2.3 to 2.6 millimolar. Inside the cell, it's less than 100 nanomolar, and in some kinds of cells, as low as 10 nanomolar. Okay. In the intercellular stores, it's about 100 micromolar. And that low, that amazingly low, really, intercellular calcium concentration in the face of very, very high concentrations outside the cells and in the stores is critically important for cell survival because calcium signaling is that important. So calcium signaling requires precise internal versus external calcium balance. Okay, so this sets up a problem in physiology. On one hand, we've got the skeleton, and that needs a lot of calcium to form and to remodel. But calcium is also absolutely critical for cellular function. And without it, you wouldn't have a functioning nervous system or a functioning heart or things like that, which are kind of important. Even if you get outside those ranges, the nervous and cardiovascular systems cease to function. So how are these physiological needs balanced? So first we have to look at how calcium is distributed around the body. So in the total extracellular fluid of our 150-pound average individual, there's about 1,000 milligrams of calcium. Now bone has about 1 million milligrams of calcium, or about a kilogram. All right, so 99.9% of calcium is stored in the bone. Okay, now the kidneys reabsorb calcium, and calcium is taken up in the diet, and on average about 1,000 milligrams a day, although the most recent standard for minimum daily requirement I think is 1,200 milligrams. Okay, that's taken up into the body through the GI, and on average only about half that makes it into the extracellular fluid. Okay, and about 325 milligrams is eliminated. So that means on an average day, about 825 milligrams of calcium ends up in the feces. So, you know, when I put these things together, I go look for these icons and stuff on Google. And if you type in feces and look for icons, it's just astonishing how many hits you get. So, which caused me to wimp out and do the toilet. All right, now of course the kidneys are constantly recycling the extracellular fluid. And so about 1,000 milligrams a day of calcium will travel through the kidneys, of which on average about 9,825 milligrams are reabsorbed, leading to an excretion of 175 milligrams, which balances out very nicely with the plus 125 taken in through the diet. And what that means is bone should be balanced. Okay. So 180 milligrams a day is acquired into the extracellular fluid through resorption, and another 280 goes into the bone through formation. Phosphate then provides the ionic balance for the calcium. Okay. Now the distribution of phosphate is a little different than calcium. There's about 600 milligrams in the extracellular fluid, and about 1,000 milligrams exist in soft tissues in the body, and that's because there's so much phosphate in DNA and RNA. Okay. About 600,000 milligrams in bone. Again, it's taken in through the gut and reabsorbed in the kidneys. Daily, most people take in about 1,400 milligrams of phosphate, of which about 1,100 gets absorbed, 200 milligrams gets eliminated, with a net of 500 milligrams out the GI, about 7,000 in the kidneys, 6,100 milligrams is reabsorbed, leading to about 900 milligrams being excreted through the kidneys. So for those of you who are interested in environmentalism, one of the coming problems in the world is that we're running out of rock phosphate, which is critical for making fertilizer. And the most obvious source for phosphate for fertilizer is the nearly 1 gram of phosphate a day every person on the planet is excreting. And all we need is some good way to recover it. But I suspect most of you will live long enough to see a day when at least some of your urine is recycled for fertilizer. That's a happy thought. All right. And again, bone is balanced. All right. 210 milligrams for formation, 210 milligrams for resorption. Okay. So calcium homeostasis is maintained by 3 hormones. Okay. The 3 hormones are parathyroid hormone, calcitriol, which is the active form of vitamin D, and calcitonin. Their regulation is based entirely on the concentration of calcium in the extracellular fluid. In combination, they control the intake versus elimination ratio in the GI, renal reabsorption versus excretion, and bone resorption versus formation. There's no CNS involvement here. Okay, this is far too important a system and far too delicate a system to have any central involvement. It has to work or you die, pretty much. The first hormone is, and probably the most important one, is parathyroid hormone. Parathyroid hormone comes from the parathyroid gland. So here's the thyroid gland on each side of the pharynx. Okay, and there are these 4 little kidney bean-shaped things sitting on top of, or actually behind, the thyroid gland. If we do a cutaway, this is histologically what they look like. Parathyroid hormone comes from these chief cells. There's also these larger oxyphil cells, and it's not clear what they're doing. Oxyphil, I think, is the stain that was used to identify them. And this little thing here is a capillary. So the biosynthesis of parathyroid hormone is pretty typical for a peptide, which is what parathyroid hormone is. Okay, so pre-pro-parathyroid hormone is 115 amino acids. Okay. It is processed down to an 84 amino acid mature hormone. But interestingly enough, only the first 36 amino acids of that hormone are active. So if you cut down parathyroid hormone and inject it into someone, you get the full activity with just those 36 amino acids. Its expression is regulated directly by calcium. It's by its storage in vesicles and its transcription. Okay. So the parathyroid hormone receptor is coupled to both G-alpha-S and G-alpha-Q. In the kidney, it stimulates calcium reabsorption and decreases phosphate reabsorption. It also stimulates synthesis of the active form of active calcitriol in the kidney. In the GI, it also promotes calcium intake or uptake. And in the bone, it's responsible for osteoclast activation or stimulates osteoclast activation, which ends up increasing calcium resorption from the bone to increase the levels of calcium in the blood. Now, the parathyroid hormone, the way the chief cells work to release parathyroid hormone, is really one of the more amazing things in mammalian physiology. So in this diagram, we've got a chief cell, okay, that releases parathyroid hormone. And chief cells have a receptor on their cell surface called the calcium-sensitive receptor. One of the real miracles of biochemistry, it can sense these tiny concentration differences in which free calcium has to exist. So the calcium-sensitive receptor is what will control, which will regulate release. Now, when the calcium concentrations in the extracellular fluid are low, just 1.1 millimolar or a little below, these receptors are unoccupied. Okay, so that under these conditions, there is a constitutively active adenyl cyclase. That means it's on all the time, all right, unless it's interfered with somehow, making cyclic AMP so that there is always, or under these conditions, an active protein kinase A. And that active protein kinase A is responsible for a transcriptional program that increases parathyroid hormone transcription and stimulates the release of parathyroid hormone. Now, when calcium concentrations are high, the calcium-sensitive receptor is occupied by calcium. When it's bound to calcium, the G-alpha-I coupled component now inhibits this constitutively active adenyl cyclase, inactivating protein kinase A, reducing its biosynthesis and its exocytosis. The calcium-sensitive receptor is also coupled to G-alpha-Q. When calcium's high, phospholipase C is activated, giving diacylglycerol and IP3. IP3 then activates calcium release. That calcium then, along with the diacylglycerol, activate protein kinase C. Okay. Now, protein kinase C, along with the high cytoplasmic calcium, inhibit parathyroid hormone release. All right, so I've been beating on this whole time that PKC and calcium stimulate release. This is the one case where they do not. Okay? So it's ever been, so I think this is a slide on top, way back in topic 2, I showed you this slide. Calcium release. And that's been, I've been beating on, and for every other system, this is true. This is the only exception. Okay? Where calcium, usually calcium activates secretion, but it's not true in sheath cells. Okay? And in a lot of ways this makes sense. I tried to explain this a couple different ways. We'll try this one. So that you have a very high concentration of calcium outside the cell. Even when calcium's relatively low, it's still very high compared to what is supposed to be in the cytoplasm. Okay? If there's any sort of accidental leak or compromise of calcium when calcium concentrations are high, if calcium then stimulated release of parathyroid hormone, it would increase the calcium concentrations in the extracellular fluid even higher. Okay? And that's potentially fatal. All right? So this release under high calcium concentration pretty much has to be prevented at all costs. And so it makes sense that a system has evolved that prevents calcium and actually causes calcium to inhibit parathyroid hormone release. The second hormone is calcitriol. It's a steroid. Okay? So it's made from cholesterol. This time it's made from 7-dehydrocholesterol. Okay? We commonly call this steroid vitamin D. Okay? And vitamin D, what is active in our bloodstream, is 1,25-dihydroxycholecalciferol or 1,25-dihydroxyvitamin D3. And this is the structure. And you can see it's made up of opening up this B ring. So here's the A ring and the C and D ring. So although on first glance the structure doesn't look that much like a steroid, it is derived from steroids. I typically abbreviate this 1,25-dihydroxy D3 with a 2 here like this. And that's just because the name calcitriol is an awful lot like calcitonin. And it's best not to get them confused. Okay. So its biosynthesis is unique among steroids. Right? So this is the one exception. The pathway I showed you for cortisol and I'll show you for androgens and estrogens does not apply here. This is the one steroid that has its own biosynthetic pathway. But its other properties should be pretty familiar to you by now. So calcitriol synthesis starts in the skin with 7-dehydrocholesterol, which upon exposure to sunlight forms cholecalciferol or vitamin D3. Okay, this is inactive, but this is the precursor. Okay, this is why you need to go outside and play and get exposed to the sun to prevent calcium issues. Okay. Now, as a steroid, it's free-floating through the bloodstream. And it can find itself in the adipose tissue where it can actually stay a long time. So adipose tissue can absorb a lot of vitamin D3 and serves as kind of a reservoir. In the liver, We can also take it up through our GI tract if you're taking vitamin D3 tablets. In the liver, it's converted to 25-hydroxy D3, which is the immediate precursor for vitamin D. Okay. It can then travel to the kidney, where it's then converted into 1,25-dihydroxy D3. And here's the 1 position. And parathyroid hormone is required to stimulate the enzyme here that converts 25-hydroxy D3 into 1,25-dihydroxy D3. In the absence of parathyroid hormone, 25-dihydroxy D3 is converted to 24,25-dihydroxy D3. That's inactive and that's excreted. So that's the metabolism of D3. Now 25-hydroxy D3 has a very long half-life, 25 days. And in the bloodstream, it's found at concentrations typically 50 to 150 nanomolar. The active form of vitamin D3 has a much shorter half-life, only about 6 hours, still pretty long, and is found at much lower concentrations, about 1,000 times lower on average. So calcitriol helps to preserve ECF calcium. So it travels through the bloodstream attached to a binding protein, because it's a steroid, you'd expect that. And it signals like a steroid. There's a vitamin D3 receptor that's part of the nuclear receptor family that the cortisol receptor and estrogen receptors belong to. Okay. It associates with another subunit and binds to the vitamin D3 response element in nuclear DNA. Okay. Now it's synergistic with parathyroid hormone in that it increases calcium, the transcription of calcium transport proteins. In the GI tract, it increases calcium intake through the lumen. In the kidney, it increases calcium reabsorption. And in the bone, it stimulates osteoclast formation. Okay, so that's how it helps promote calcium resorption. Also in the parathyroid, it decreases transcription of parathyroid hormone. Okay, and that always fouls people up, but we'll get to why that makes sense in a second. But it increases transcription of the calcium-sensitive receptor, and in general is needed for maintenance of the parathyroid chief cells. The third hormone is calcitonin. Okay. So this is the hormone that's antagonistic to parathyroid hormone and 1,25-hydroxy D3. It's peptide hormone, and it's made in the thyroid gland C cells. Okay. So go back to our thyroid here, and there is the C cell out here, next to the follicular cells, and that's where calcitonin is made. High calcium in the extracellular fluid stimulates secretion of calcitonin. It's also regulated by the calcium-sensitive receptor, but in a way that's opposite that of parathyroid hormone. So as you might guess, in the C cell, calcium-sensitive receptor is coupled to G-alpha S and G-alpha Q. Now this is overall a pretty weak signal. And it's controversial how important it is in humans. Okay. The calcitonin receptor is a G-alpha S coupled receptor. Okay. And it's most active in the bone, where it helps to depress osteoclast activity. And in fact, the classic way to identify osteoclasts on the bone is by doing a stain for calcitonin receptors. So parathyroid hormone, calcitriol, and calcitonin regulate calcium homeostasis. Parathyroid hormone and calcitriol work in the kidneys, the GI, and the osteoclasts. Calcitonin strictly the osteoclasts. Okay. So the molecular mechanisms in the GI and in the kidney are pretty much the same. So we'll just go over the one. We're just going to go over kidney. The osteoclast mechanism it's not so much unclear as incredibly complicated. And I've tried to get it in, and it never works. So we're just going to skip it. It's one of these things I'm just not going to be able to get to with the time allotted. Okay. And we go back to one of our original principles here that is going to be necessary to keep in mind to understand how these hormones are working, is that calcium and phosphate in the extracellular fluid are reciprocally regulated. When one goes up, the other has to go down. Okay. So this is in the kidney, where parathyroid hormone and vitamin D3 regulate calcium reabsorption. Okay, so here's the lumen down here, okay, and the extracellular fluid on the basolateral side. The bloodstream's up here. Okay, so when calcium is high, there's a sodium phosphate cotransporter that reabsorbs phosphate, and then through mechanisms that are unknown, it gets into the extracellular fluid and keeps phosphate concentrations high in the bloodstream. Now, when calcium's low, parathyroid hormone is released, and it promotes the synthesis of vitamin D3. The vitamin D3 then activates a transcriptional program that causes transcription of a calcium channel and of a sodium-calcium exchanger and calcium ATPase. Parathyroid hormone then, through protein kinase A, causes the transport of this calcium channel to the cell surface, okay, as well as the transport of the exchanger and the ATPase to the cell surface on the basolateral side. So this apical calcium channel then allows calcium to be reabsorbed from the lumen, where it's then pumped or transported out of the cell by either the ATPase or the sodium-calcium exchanger, increasing the calcium in the blood to compensate for the low levels. Now, I've said a couple times that the phosphate's reciprocally regulated with the calcium. And so parathyroid hormone also then inhibits transport of the sodium phosphate cotransporter to the cell surface, decreasing the reabsorption of phosphate in through the lumen. And the GI mechanism is not exactly like that, but it's close enough, so we probably don't have to worry about it. So why are calcium and phosphate reciprocally regulated? All right, so here is a bone, and this little circle here is meant to symbolize hydroxylapatite. Okay. Here's our osteoblast and osteoclast, and here, of course, is a kidney, bloodstream. Okay. And this reciprocal regulation is a consequence of mobilizing calcium from the bone. So when calcium concentrations are low, parathyroid hormone and vitamin D reabsorb the calcium and the phosphate from the bone. But they come out in a fixed molar concentration. Okay? Because you're dissolving a mineral, you can't just take some calcium and leave the phosphate behind. You have to take them both. Okay? So calcium and phosphate both increase in the bloodstream. Okay? So they're mobilized equally. The problem is that this leads to excess phosphate in the extracellular fluid. Okay? So that excess phosphate then is excreted. Again, under the influence of parathyroid hormone and vitamin D, calcium is more efficiently reabsorbed and phosphate is more efficiently excreted, okay, thus reducing the phosphate concentration in the extracellular fluid to an acceptable level. Now, when calcium is high, under these conditions, we're favoring bone formation. Bone formation requires set molar concentrations of calcium and phosphate to make the hydroxylapatite. Okay? But now phosphate's too low in the extracellular fluid. Under these circumstances, the parathyroid hormone is very low, vitamin D conversion is inhibited. Okay? And so just then the constitutively then, more phosphate is excreted, and more calcium is excreted, more phosphate then is reabsorbed, giving you equal molar concentrations in the bloodstream. Okay. And then partially under the influence of calcitonin then, to inhibit the osteoblast then favor formation, giving you hydroxylapatite in the bone. Okay. So that's why they have to be reciprocally regulated. So there's a fairly complicated picture of reciprocal control here. It's really very elegant, and essentially it's all on autopilot, assuming everything's normal. Okay, so we start down here in the parathyroid cell, in the parathyroid gland, when calcium's low. Under those circumstances, parathyroid hormone is released from the parathyroid gland. It binds to receptors on the kidney where it converts 25-hydroxyl D3 into active 125-hydroxyl D3. These two hormones then cause calcium to be reabsorbed and favor phosphate excretion. Parathyroid hormone in the osteoclast then inhibits or stimulates the osteoclast as well as vitamin D, and that causes more calcium and phosphate to be reabsorbed from the bone, increasing calcium concentration and phosphate concentration because the phosphate is kept down through excretion from the kidney. Parathyroid hormone and vitamin D3 also, similar to in the kidney, promote recovery of calcium from the diet or intake. Now, under these circumstances, vitamin D inhibits parathyroid hormone. Okay? And this is part of this homeostasis thing. You don't want parathyroid hormone to stay high forever. Right? It's out for a while, it has a short half-life, and pretty soon you want to down-regulate it because this problem will be taken care of. Right? So vitamin D3 inhibits transcription of parathyroid hormone. All right? So at this point, calcium concentration should be increased. Now, if calcium is high, above 1.3 millimolar, the calcium-sensitive receptor now is occupied, inhibiting release of parathyroid hormone. The loss of the parathyroid hormone signal then causes production of the inactive 24-25 vitamin D3, which is excreted, causing loss of the vitamin D3 signal, or decrease of it, okay, and increased recovery or decreased recovery of calcium and increased recovery, reabsorption of phosphate to equimolar amounts in the bloodstream. Calcium intake is decreased, and the calcium-sensitive receptor here in the thyroid, the C cells, causes the release of calcitonin. Calcitonin then, with its receptors on the osteoclast, inhibits reabsorption, and by inhibiting reabsorption, favors formation, causing calcium and phosphate to be removed from the bloodstream. Okay. In the absence of vitamin D, that transcriptional inhibition is lost, and now parathyroid hormone is actively transcribed, getting it ready for the next time when it has to regulate calcium concentration. Again, this has to happen fast. Okay. So I've emphasized that bone's a dynamic tissue, but it's even more dynamic than I let on. There's a lot of levels for control of bone and calcium homeostasis. The one I've given you is the acute one, is the most important. Okay. There are many diseases that involve bone, and I can really only scratch the surface of this. The only one we're going to consider at all is osteoporosis. So there are other interactions between bone and the endocrine system, especially in the reproductive axis. We talked a little bit about growth. Okay. There's also really important interactions with long-term energy storage, which I don't have time to get into at all. Okay. And these are more responsible for bone growth and maintenance than calcium homeostasis. Okay, so these are the hormones that stimulate bone growth. Okay, and there's a long list. You don't have to memorize it. Just be aware it's there. Okay. And for some reason, evidence is mounting that TSH in the bloodstream inhibits bone formation for reasons that are unclear. But cortisol inhibits bone formation as well. Okay, and in fact, in men, we'll talk about osteoporosis in a minute. In men, the number one cause of osteoporosis is glucocorticoid intake. This is a side effect of glucocorticoids. Okay, and that brings us to osteoporosis. Okay, so osteoporosis is a decrease in bone mass leading to fractures. Okay, so if you take a normal bone and dissolve the protein in the cells and everything out of it, and just look at the mineral matrix, it looks something like this. Okay, and an individual with severe osteoporosis will have a much more open structure. Almost 10% of the United States population is afflicted at least a little bit by osteoporosis. Although in an awful lot of these people, it's asymptomatic, it hasn't caused a fracture. And this population is made up mostly of elderly women. And the root cause of osteoporosis is a maladaptation in the osteoclast-osteoblast activity ratio. So that reabsorption is favored over formation. Right. Now, osteoporosis is often thought of in terms of calcium deficiency. Right. And it's true that sufficient calcium and vitamin D are certainly critical for maintaining this ratio. Okay. However, and exercise is also another important stimulus for maintaining this ratio. Formation is, there are stretch-activated receptors where the tendons attach to bone, and so weight-bearing exercises activate those receptors and cause and stimulate bone formation everywhere. So that an exercise, say exercising your bicep, will actually increase bone formation in your leg. Okay. But serious cases, significant cases of osteoporosis can't be reversed just by increasing calcium and vitamin D3 in the diet. Okay. There needs to be some sort of pharmacotherapy that favors formation. Okay, to see why that is, let's go back to our balance diagram. Okay, and under normal conditions, you have a net plus 175 from the GI, which absorbs about 50% of the available calcium, a net loss of 175 milligrams from the kidneys, and bone should be balanced. Okay, let's reduce the amount of intake by 500 milligrams. Now, I'm making these numbers up. I looked really hard for actual numbers and couldn't find any. But so the actual numbers may be off a bit, but this is the idea. Okay, so there's no reason to memorize these numbers. I'm just trying to give you an idea what happens. Okay, so in this chart over here, we've got our 500 milligrams. Okay. Under these circumstances, the GI will probably increase the amount it intakes. Okay. So that, let's say it goes up to 70%. All right. So that less is coming in, but it's being absorbed more efficiently. So we're going to reduce it by 150 milligrams. Okay. So now it's 350 in, 325 out for plus 25. The out's not really regulated. So the net intake's only 25 milligrams compared to 175. Now the kidney will also upregulate its reabsorption of calcium under these circumstances. Okay. So let's say we're only losing 125 milligrams in the urine, okay, for a net loss of 50. Or we're conserving an extra 125 milligrams in the urine for a net loss of 50 milligrams. Okay. So now we're still down by 25 milligrams, which doesn't seem like a lot. And of course, bone's there. This huge amount of calcium in bone is there to serve as a reservoir. But it's no longer balanced. So that to maintain this 1,000 milligrams of extracellular fluid, an extra 25 milligrams has to be reabsorbed out of the bones. Okay. All right, so that's what the deficit's made up. Now, this doesn't seem like a lot. But over a decade, that's a loss of 90 grams of bone mass, almost 10% of the total. Okay, so you can see how chronic calcium deficiency, and I think this is actually a pretty optimistic scenario. Under chronic calcium deficiency, you can see over time, it will really cause a compromise, or vitamin D deficiency, in the amount of bone mass. Okay. So osteoporosis and prevention treatment. The first time I talked about this, it was a disaster. People started running home and telling their grandmothers to quit taking their vitamin D. It didn't do any good. Don't do that. Because I learned my medicine out of a physiology textbook, and that's just a bad idea. In diet for prevention, it's important to have sufficient dietary calcium to saturate the uptake mechanisms, okay, and sufficient vitamin D to maximize its conservation effects. Okay. But those can only be so high. Okay. Once there's been net calcium loss, insufficient calcium and net calcium loss can be caused by insufficient vitamin D or calcium, or some underlying condition like kidney failure, where the kidney is incapable of reabsorbing calcium at the level needed. But once the loss is significant, the calcium and vitamin D can decrease the rate of the loss by readjusting the diet. But you're not going to reverse it because of the whole homeostatic mechanism we talked about. You simply can't move the calcium concentration in the bloodstream very much. Okay, so it's hard to reverse a long-term deficiency just through diet. In fact, it's probably impossible. So this is necessary. Sufficient vitamin D and calcium are necessary, but not sufficient. Okay? What's needed then is some sort of pharmacological treatment, usually, that favors bone formation over reabsorption. So weight-bearing exercises help. But there's a lot of pretty good drugs out there that help. And it's early days with them. I think the research is sort of optimizing their dosages and such. And there's another generation of drugs coming down the pipeline that I think will help a lot. But they all work on this kind of strategy. Oop. Okay. So this ends the material that's going to be covered on exam 1. All right? Now, I would suggest you not go away at this point. Unfortunately, reproduction is probably the most complicated thing we're going to go over. The loops and the feedback mechanisms and stuff are very complex. The reason I save it till last is because up until you really have to see this stuff in simpler systems to have any idea at all what's going on. And it seems like it's going to be a year before you take the final. And it's best you look at it now and kind of get it in your head while you understand all these things. And then you can, for the final, you can just go back and review it. And I'll put out some material before the final that'll help. Okay, so let's do, what time is it? Oh, we got time for 3 questions. Why don't we do that? Maybe 3, we'll see. Okay, I'm just going to make it visible now and activate it once you've had a chance to read it. How's that? Here we go. Slow internet day. All right. So calcitriol, the active form of vitamin D, is a signaling mechanism most like that of which of the following? So, come on. There. Okay, so you should be able to submit. We'd like to see the question, please. Come on. Ah, here we go. All right, so go ahead and submit your answer. All right, a few of you are right on this. Either that or you're one of these people who has 3,000 texts a month and can just fly. Deepen your notes. You know, notes aren't available on the test. Unless you figured out some system. I'm sorry, what? Any of you? Okay, so talk to the top hat people. So they when there's a problem, they've got the technical support. That's one of their advantages, and they have been able to get people on. Okay. So first try top hat technical support, and then we'll. So some people have been able to get on. Well, some people, yeah, have been able to get on. Okay, so it's still compiling submissions. So maybe it will. All right. Was this an easy one or a hard one? Easy, hard, so, all right. All right, so it still seems to be compiling answers for whatever reason. All right, so I hope those of you who said glucose understood glucose isn't a hormone. It doesn't have a signaling mechanism. And you may have misread it as glucagon. You just pay attention. And the rest are all proteins. Thyroid hormone is not a steroid, but it's something like a steroid. And so thyroid hormone and vitamin D go together. And that's part of that sorting of hormones I think you should go through. So in signaling mechanism, thyroid's like a steroid, vitamin D is a steroid, and I expect you to be able to put the two together. But yeah, I think 80% is about what last year's class got. All right, we're going up here. We're down. All right. Hey, whatever. I don't care. That's very clever. All right. I can never remember if those buttons mean that it's what will happen if you press it or what's happening now. All right. Go back. All right, items. Here we go. All right. All right, let's do another question. This is an easy one, I think. If it will come up. Oh, here we go. What percentage of the body's calcium is present in the bones? This is one of those questions I sometimes put in the test because I get worried that the grades are too low. And then a third of the class misses it and I get depressed. And then I remember it's your fault and I'm not so depressed anymore. All right. So how many got this one right? Should be almost, some of you are not very confident. All right. Yeah, so it's 99, it's the vast majorities, like 99.99%. So it's 1,000 milligrams, or about a kilogram in the bone, and only 1,000 milligrams, or 0.01 grams in the extracellular fluid. Okay? All right, so let's just do two. If we have time, maybe we'll come back and do a third one. Okay, so this is male reproductive endocrinology, and that, for reasons I hope you'll see, leads us logically into sexual differentiation in the fetus. Okay. So we'll cover the gonadotropins, which are the HPA axis hormones, synthesis and regulation of male sex steroids, so a little bit of female, sexual development during gestation, and then we'll take a little time at the end to go through disorders of sexual differentiation. So the endocrine system dominates reproduction. We all know this at some level, I hope. It's required for fetal differentiation at the beginning of life. It regulates the timing of reproduction, both through monthly cycles in women and through the timing of puberty. It's responsible for initiation and maintenance of pregnancy. And one interesting feature of it that most people don't realize is that the hormones involved, for the most part, are common to both sexes. There's heavy involvement of the HPA axis and gonadal steroids. There are gender differences in the steroids, but they're mostly in the amounts of the steroids, not the identification, not the identity. And also there's big differences in the way they are expressed in the timing of their expression. But their similarities are important. The anatomical organization of gonadal steroid synthesis is pretty similar in men and women. Okay? And many of their functions are analogous. Okay, so hypothalamic and pituitary control is common to both sexes. Okay? So from the hypothalamus, we have gonadotropin-releasing hormone. It's a 10 amino acid peptide. It has a G-alpha Q coupled receptor. It's released in pulses. In men, these pulses come once every 1.5 to 3 hours, and that timing can be irregular. Okay. In women, adult women, those pulses come once an hour on average. It can be as short as a half hour or as long as about 1.5 hours, but it's once that timing is established, it'll be very regular throughout her lifetime. Gonadotropin-releasing hormone targets the gonadotroph cells in the anterior pituitary. From the gonadotroph cells come two hormones, collectively known as gonadotropes: luteinizing hormone and follicle-stimulating hormone. They both bind to G-alpha S coupled receptors. They are glycoprotein hormones. They contain a common alpha subunit of 90 amino acids. That's the same alpha subunit found in thyroid-stimulating hormone. However, each has a unique beta subunit that's glycosylated. And that glycosylation is required for their activity. It won't bind the receptor correctly if the beta subunit isn't glycosylated. So GnRH from the hypothalamus stimulates FSH and LH release. Okay, so, oops, there we go. Sorry. All right, so gonadotropin-releasing hormone binds to its receptor. Okay. Activates phospholipase C, giving diacylglycerol and inositol triphosphate. The inositol triphosphate promotes intercellular internal calcium release. That calcium then opens another calcium channel, further increasing the calcium inside the cell. Again, I'm not that concerned, you know the difference. The calcium and the diacylglycerol activate protein kinase C, Protein kinase 3C through a transcriptional program causes transcription of LH and FSH, and the calcium promotes release of LH and FSH. Okay. And this mechanism is common to both men and women. It doesn't vary at all. Feedback varies. So sex steroid synthesis. So we know androgens, like testosterone, dominate in males, while estrogens dominate in females. And most people who know a little know that much. But it's really important to keep in mind that both sexes require both androgens and estrogens for normal development, reproduction, and behavior. So in both sexes, sex steroid synthesis has a common pathway. So we've got cholesterol esters, steroid acute regulatory protein, cytochrome P450 side chain cleavage to pregnenolone, progesterone, 17-alpha hydroxylase. Androgen synthesis requires, and estrogen synthesis require the 17 to 20 desmolase to give DHEA and androstenedione. And then a 17-beta hydroxysteroid dehydrogenase to give androstenediol and testosterone. To get estrogens, you need aromatase. Okay. Now in this pathway, LH, as we'll see, stimulates steroid acute regulatory protein and the side chain cleavage and the 17-alpha hydroxylase, as well as the desmolase. Aromatase, however, is stimulated only by FSH. Now, throughout this topic, when I brought up estrogen, so there's nothing really called estrogen. More correctly, we should be calling them estrogens. And that's because there's 3 estrogens, only 2 of which are found in males, while one of them is only found in pregnant women. Okay. They go as estrone, which is E1, and estradiol, which is E2. Estradiol is more abundant and more potent, so normally we should probably use estradiol. But when you see estrogens plural, it's some mix of these two, usually. Okay. And E2 is the most important in adults. Okay, so to synthesize androgens and estrogens, to look at testicular anatomy, so this is a cutaway of a testicle. Okay. If you take this little cutaway here inside the lobes, you get the seminiferous tubule. This is a cross-section of the seminiferous tubule. Okay. It has 2 important cell types. Here's the developing sperm, the lumen. Okay, these are the mature sperm. This is spermatogonium, a developing sperm cell. This is a Sertoli cell, and this on the outside is a Leydig cell. Okay. The Leydig cells make the testosterone. Okay. They have LH receptors. Sertoli cells nurture developing sperm. They have FSH receptors. FSH then is responsible for synthesis of estrogens, growth factors required for spermatogenesis, and synthesis of the androgen binding protein for circulating testosterone. Okay, so LH regulates testosterone synthesis in the Leydig cells. G-alpha S-coupled receptor. It activates adenyl cyclase, giving active protein kinase A. Okay. Now, to complete the program, protein kinase A directly stimulates steroid acute regulatory protein. To complete the program, growth hormone and prolactin are both required. Okay. The combination of luteinizing hormone, growth hormone, and prolactin then are responsible for a transcriptional program plus PKA, right, PKA, that increases synthesis of STAR, side chain cleavage enzyme, the 17-alpha hydroxylase, and the 1720 desmolase, resulting in testosterone. The testosterone then diffuses through the basolateral membrane into the Sertoli cells, where it creates very, very high localized concentrations of testosterone in the Sertoli cells, which is absolutely required for spermatogenesis. And that's also the source of circulating testosterone in men. FSH then induces Sertoli cells to support spermatogenesis. Okay, so we have our testosterone here. Okay. It directly interacts with the developing sperm. FSH then results in active protein kinase A, which gives us a transcriptional program that results in synthesis of androgen binding protein, growth factors, and more than 100 other proteins required for spermatogenesis. Importantly, we also get activation or increased synthesis of aromatase. This is an enzyme name I want you to have memorized. And this enzyme converts testosterone to estradiol. The estradiol then diffuses back across the basolateral membrane along with the growth factors. It's responsible for a transcriptional program critical for Leydig cell maintenance and proliferation. And in fact, every place you see estrogen, you see cell growth and proliferation. Estrogen is effectively a growth factor. There's then feedback from the developing sperm, which provides additional growth factors to help maintain the Sertoli cells, along with the estrogen and its internal growth factors. So peripheral tissues are also involved in sex steroid synthesis. So we have testosterone being made in the Leydig cells, which circulates, some of it circulates. And in this case, testosterone, we've already seen how testosterone is a prohormone for estrogen. Okay, so estradiol in men is made in Sertoli cells, where it supports sperm development. But that's a localized source. Okay? There's not enough of that estrogen to really find much in the circulation. Okay? Circulating estrogen in men comes from the adipose tissue. Okay? Adipose tissue, then you should be able to guess, has aromatase, okay, which is not under a lot of regulation. And so this is the source of circulating non-localized estrogen in males. Okay? It occurs in women as well, but it's nowhere near as important. In men, it has both metabolic and behavioral roles. There's also 5-dihydrotestosterone, or 5-DHT. That's made in peripheral tissues by a 5-alpha reductase, which gives 5-DHT. Most 5-alpha reductase is in the skin and in the prostate gland in men, where it's responsible for the development and maintenance of many secondary sexual characteristics. Okay, so the feedback regulation of gonadal steroids in males is pretty standard, I hope. You think of it that way. So the stimulus is really, I put time, there's environmental stimuli. The gonadotropin-releasing hormone stimuli is pretty complicated, and there's no one huge important factor. So it's more or less tonic. So that then causes synthesis and release of LH and FSH. Cortisol inhibits LH and FSH release, or synthesis. Growth hormone stimulates it. Luteinizing hormone then, along with prolactin and growth hormone, results in testosterone synthesis. Testosterone then is required for nurturing developing sperm. FSH, along with growth factors from the sperm and the Sertoli cells, plus estrogen and androgen binding protein, are all made in the Sertoli cells. Those growth factors in the estrogen help the Leydig cells and the Sertoli cells to be maintained and proliferated. Estrogen is made in the adipose tissue, DHT in peripheral tissues, and the circulating testosterone then is involved in negative feedback for GnRH and LH and FSH. Testosterone is also important for behavioral inputs in men, as well as estrogen. Okay, so over a lifetime, the levels of testosterone in men change considerably, as well as FSH and LH. Okay. So during gestation, there's a pulse of gonadotropins and testosterone. And there's also, as in infants have, right after they're born, have a quick little spike in LH, FSH, and testosterone for reasons that are unknown. Okay. Around age, somewhere around 10 to 12 usually, there's a little spike in the gonadotropins, here are the dashed lines. And that heralds puberty. Okay. And at that point, testosterone levels start to increase, okay, along with the gonadotropins, until men get to be, and this can vary a lot, somewhere between age 56 and 65, 70, 80, somewhere in there. And what will happen is that testosterone levels will start to decrease and gonadotropin levels will start to increase. And it should be clear to you why that would be. Maybe it's a test question. Okay. Androgens and estrogens are in both sexes. Okay. So in males, and to a lesser extent females, steroids have other functions here. So testosterone then is important for development of internal genitalia. It's uniquely important for men developing a deep voice. It has metabolic effects. In the liver and adipose, it's anabolic for lipids, and it's also anabolic for proteins and induces IGF-1 synthesis. DHT is uniquely required for development of the external genitalia, beard growth, sebum formation, and prostate function. And along with testosterone, is required for general pubertal development. Estrogen in men, then, is critical along with testosterone for bone development. Okay. Uniquely, it's catabolic to adipose lipids by stimulating synthesis of hormone-sensitive lipase and depressing synthesis of lipoprotein lipase. And it also induces cell proliferation in many systems. Okay. And along with the testosterone, it's important for behavior and sperm production. These metabolic effects are true of both men and women. So this graph shows the relative concentrations of androgens and estrogens in both sexes. So the solid bars are men, the cross-hatched bars are women. Okay? So we've got estrogens up here. So this is the log of the hormone concentration. So you can see in men, circulating estrogens are quite a bit lower than in women. DHEA, dihydroepiandrosterone, and dihydroepiandrosterone is at about the same levels in both men and women. There's not as much difference in DHT as you might think, although most DHT doesn't circulate. Peripherally, there's quite a bit more testosterone in men than women. And if you just sample from the testicular vein, you can see you really have a very, very high level of testosterone. This is what's required for gonatogenesis. Okay. So in women, DHEA is most important, and that comes from the adrenal gland. It's considered a weak androgen. And the reason it's weak is because it has a low affinity for the androgen receptor. Okay. And women don't have that much of the androgen binding protein. That depresses free concentrations even further. Right? In both sexes, most DHT does not circulate. Okay. And you can see, again, I pointed this how much higher, the almost 100-fold higher in the testicles testosterone than in the peripheral bloodstream. So let's go back to the adrenal gland then. Okay. And the androgens are synthesized in the zona fasciculata. Okay. So we have cortisol up here. This is the zona fasciculata. I'm sorry, I've got this messed up. Okay. In the zona fasciculata is where cortisol is synthesized. Okay. And it is rich in the 3-beta dehydrogenase, but has very little of the 1720 desmolase. Okay. So the pathway to cortisol is favored, but you get very, very little androgen. So you get, the result is glucocorticoid synthesis. Now in the zona reticularis down here, there's a lot of the 1720 desmolase, but very little of the 3-beta hydroxylase. Okay. So the result is DHEA synthesis is favored. Okay. Now in many peripheral tissues have the desmolase and the 17-beta hydroxylase to make testosterone, and then the 5-alpha reductase to make 5-DHT found in women. Okay. And in women, testosterone will influence behavior, and especially the 5-DHT is important for some secondary sexual characteristics. Okay. It also has those metabolic effects I mentioned before. So sexual differentiation in utero. So that's about all we need to know about endocrinology in men and where androgens come from in women. Okay? And this leads us to sexual differentiation in utero. Okay? And the whole point here is to know that gender differences are established by the endocrine system during development. Okay? When you learn this, you think of it in terms of genotype. But it's not the genotype, it's the endocrine system. So we have men and women, and there's a perception versus reality issue here. So we all know about the genetic part. We've got X, Y, and XX. And these traits are invisible. You can't look at a person and tell what chromosomes they have. Okay. And these are set at the onset of fetal development. But there are also gonadal characteristics and phenotypic characteristics, things like size, facial hair, voice, The phenotype and the genotype don't have to be the same thing. Okay? Typically, we associate a very tight link, but there's a lot of ways that can be interfered with. Okay? And this phenotype gives us our appearance of male or female, and what we perceive as male and female, and the different ways we treat men and women. All that's based on endocrine factors. Okay? The fetus then starts off indifferent. So early in gestation, you can't look at a human fetus and tell whether it's male or female. Endocrine factors then determine the gonadal and phenotypic traits pretty early in development. Okay? Of course, this is under the influence of genotype, but again, it can be interfered with. So prior to 9 weeks gestation, a fetus has the potential to develop either male or female. Okay? We refer to the fetus at this stage as sexually indifferent, and it has a duct system. So here's the fetal gonads. It has Wolffian ducts and Mullerian ducts. Oops, sorry. Hit the button. Wolffian and Mullerian ducts. Mullerian ducts will lead to a female reproductive system, while Wolffian ducts will lead to a male reproductive system. Male development is determined by the presence of a protein called testes determining factor. So here's the primordial germ cells in an indifferent fetus. In this case, is an X and Y chromosome. If we blow up a map of the Y chromosome, out here, right at the end of the short arm, there's a gene. Okay? It's called the SRY gene. And it's most of the reason a Y chromosome exists. That protein, encoded by SRY, is called testes determining factor. And it's a transcription factor expressed in the fetal gonad of genotypic males. And it causes transcription of another hormone called, or of additional hormone called anti-Mullerian hormone. So anti-Mullerian hormone, or AMH, is a glycoprotein consisting of 2 identical 70 kilodalton subunits. It's part of the TGF-beta superfamily, for whatever that's worth. And the receptor it binds to is a serine threonine kinase. So these come as 2 non-identical subunits. Okay? Oh, I'm sorry. And this is expressed in fetal males, and it's key to sexual development. It's also expressed in male children until puberty for reasons that are not clear. And it's expressed in females after puberty where it's required for fertility. But again, its role is really unknown. Okay. So AMH signals through these serine threonine receptors. 2 receptors are required. They're non-identical. Anti-Mullerian hormone first binds to receptor 2, and then receptor 1, the type 1 associates. That activates a kinase on receptor 2 that phosphorylates a kinase to activate it on receptor 1. That serine threonine kinase then phosphorylates a transcription factor called SMAD, okay, which came from a combination of SMA and C. elegans, a worm, and MAD in Drosophila. MAD stands for Mothers Against Decapentaplegic. Drosophila people just love these weird names. Okay. And then that complex then activates the transcriptional program that leads to development of a male. So anti-Mullerian hormone results in a male. Its role is to cause atrophy of the Mullerian duct. Okay, so the fetal gonads express anti-Mullerian hormone, and the Mullerian duct atrophies. Then testosterone from the testes stimulate development of the Wolffian duct into the internal genitalia. 5-DHT made in external target tissues result in male external genitalia. Female development is caused by the absence of testes determining factor. So while there's a positive factor required for men, the default developmental pathway in the absence of any interference is female. So if testes determining factor is not expressed by week 9, you end up with a female. In which case, the Mullerian duct develops into the internal female genitalia, the Wolffian duct atrophies, and the external genitalia will develop female in the absence of the signal from 5-DHT. Okay. Now, this isn't all there is to it. Functioning ovaries require 2 functional X chromosomes. Okay, so there are additional factors involved. But this is how it gets started. So let's quickly compare male and female developmental pathways. So here's a normal male. In the presence of SRY, we get male gonads. Those lead to testes. The gonads also express anti-Mullerian hormone, causing atrophy of the Mullerian duct. And then under the influence of testosterone, development of the Wolffian duct into internal genitalia. The 5-alpha reductase then gives 5-alpha DH, or 5-DH, and that causes development of external genitalia. Females do not have SRY. You get female gonads. Along with another X chromosome, you get ovaries. Under these circumstances, the Wolffian duct then atrophies. The Mullerian duct develops into internal genitalia, and the external genitalia develop by default. Okay, so let's end it there, and Thursday we'll finish this up with disorders of sexual differentiation. Hey, that's okay. Saw your name. Congratulations.
Oh, thank you.
I know, but you know, when you're over in Cary, you never hear anything. I was too far away to hear you whooping down the hall.
Yeah, I know.
So, and actually, there's some, one of these days I got to come up and talk to you about that. There's something I need to, since you're now an expert on pain, I need to talk to you about something.
I got to ask you about all the chromes that you have. By the way, do you happen to have any of the